pharmaceutical. So for this, we need precision. Yeah, so how to convert just the crude herbal extract and all the paste become a safe, standardized a formulation. Yeah, so we need precision. Yeah, we need to know how it works. Yeah, at a molecular level. Yeah, so if we know how it works at molecular level, yeah, so we understand how this extract works to treat the disease. And then after that, we also need to know how to manufacture this extract or this paste, yeah, without destroying the environment. Yeah, when we talk about manufacture, so we need a supply of raw material. How to manufacture this extract, yeah. So we can do without any destroying of the environment. So for that, yeah, we must cross the bridge into advanced biotechnology. We don't use the conventional method to screen or to study the traditional medicine. So we can bridge into advanced biotechnology. So where this bridge can bring us to the next topic about the technological bridge, advanced biotechnology. So next slide, please. Okay, so now that we understand the deep history of ethnobotany. Yeah, so there is that I mentioned before. There is a lot of traditional medicine that have been used and become a foundation. Yeah, for the drug that we use now. Yeah, like aspirin, vincristine, quinine, and so on that I explained before. Okay, so now let's talk about the bottleneck. So for decades, the biggest challenge in natural product research was speed and complexity. Yeah, so because when we start the research on natural product, so we need to screen the compound, we need to isolate the active compound, and we need to know what compound is responsible for the action. Okay, so and then this research is quite the time consuming and complex. Yeah, a single medicinal plant can contain thousands of distinct chemicals. Yeah, how do we find the needle in the haystack? Yeah, so this becomes a question. Yeah, so sometimes only maybe just one compound that responsible for the effect. Yeah, so how do we find the needle? Yeah, so this is where the technology bridge comes in. Yeah, we are going to look at how modern biotech turns raw, complex traditional remedies into precise, scalable pharmaceutical innovation. Okay, next slide, please. Okay. Our first structural that we can use, we can become a bridge, the traditional, we can say a traditional medicine into a modern medicine. Yeah? So this through high throughput screening or HTS. Yeah, in the past, evaluating traditional remedies. The scientists spend months or maybe years manually testing and extract in the petri dish. In test tube. Okay, so this during the traditional approach. Okay, so during my time when I was in degree. So today there is a robot for this, through the high throughput screening. So they use robotic. So when we use HTS, we can take hundreds of plant extract, maybe that suggested from the traditional healer, place them into microplates, and let automated system test and against maybe specific diseases target, for example, like cancer or viral protein or any targeted or any other protein. Okay, so this testing in a matter of hours compared to when you do the traditional approach, maybe months, maybe years. Okay. So STS allow us to validate the biological activity of traditional medicine at unprecedented speed. Okay, so this how actually the technique or the equipment that can help us to move faster, in the nature product research. Okay, next slide, please. Okay, so but the another concern for the traditional medicine, traditional medicine rarely rely on just one molecule. It rely on the harmony of entire extract. For example, like to reduce the glucose level. So in the traditional the way how people try to reduce the glucose level, they use a cinnamon. Cinnamon. They just soak cinnamon in the glass of water, hot water, and drink the water. Okay, so we don't know which one, the compound that is responsible. But actually, the traditional, they rely on the harmony of the entire extract. So this brings us, so if in this condition, where we need all the component in extract, so this brings us to the metabolomic and artificial intelligence. So as we know that metabolomic, so this is the comprehensive study for all molecule or metabolites that present in the biological sample. So from metabolomic allow us to take a chemical snapshot of every single compound in plant. And then, after we know, after we study the metabolomic, so then we fit this massive data, massive data matrix into AI algorithm. So the AI can look at the data, so it can predict which combination of molecule is responsible for the therapeutic effect, and even show us the work together synergistically. So this the advantage of AI. This more on the prediction. Okay? Instead of reducing a plant to a single drug, AI help us understand the complex, multi-targeted physics of how traditional formula actually work. So this the technology that will help us. So from the traditional medicine, we have the data. We have all the chemical component in the extract. We have all the metabolomic data. The massive, we can say a massive data, and we also can do the AI prediction. So this become a synergy mapping. So we can identify how actually the traditional medicine work. Okay, next slide, please. Okay, so once we find the perfect molecule or compounds, or maybe the combination of the compounds that have a responsible to the activity. Okay, so we will face a major ethical and environmental hurdle. This is what we call as a supply. So meaning that from the so we know the everything about this plant. and limit supply, when come to the manufacture. Okay, so if a rare rainforest tree holds a cure, maybe the rainforest tree is rare, they can use to cure a disease, we cannot simply chop down the rainforest tree, and then harvest it. Okay, so how to do this? So through the synthetic biology can solve this problem. Okay, so how? By mapping the genetic pathway of the plant, how this compound produced in plant. So we know the mechanism of the synthesis of plant. So the metabolic engineer in this situation, they need to copy paste how the plant's chemical recipe into the DNA of the DNA of common we can say as a baker, into a DNA of microorganism, for example, E. coli. So meaning that we copy the genetic engineer will copy all the mechanism of the synthesis of the target compound, and we paste into the DNA of the, for example, yeast or E. coli. So yeast and E. coli, when they have all the information to produce that chemical, this will be synthesized in their cell. So we turn these microbes into cellular factory. So this, for example, E. coli. So E. coli have this kind of information. So they become factory to produce this chemical. Instead of harvesting the endangered flora, so we can brew the pure active medicinal compound in sustainable industrial bioreactor. So here we use bioreactor. So now the famous production using this technique is the production of insulin. We use E. coli, and then we produce insulin through the bioreactor. Okay, so this actually how this not only for insulin, but also we can use for other chemical where we deal with the endangered species. Okay, next slide, please. Okay, so here, so we can see here, the traditional knowledge, so, like I said, this become a foundation of natural product discovery. Okay, so from traditional knowledge, they provide the empirical proof. So where the remedy works within the human context. Knowing that a plant works is only half battle. So to turn the crude herbal extract or tea into a self-stabilized global distributed pharmaceutical, we need precision that I mentioned before. Okay, so we need advanced biotechnology. So where this will help us to provide the sustainable sources. Especially for if this this plant is endangered species. Okay, so next slide, please. Okay, so now I try to bring all of you the theoretical and then how actually the evidence. Okay, So we have established the theoretical bridge of biotechnology, and then let's look at the actual evidence. So this part, I will explain three proof of concept through the real world cases study. Okay, so we are going to look at the three distinct sample. So where these three distinct sample actually the merger of the ancient observation and modern molecular biology. So they not just yield a drug. Actually, this radically redefine modern clinical therapy and save millions of lives. Okay, so next slide, please. Okay, so our first case study. So this I can say this case is the absolute gold standard of integration. So the discovery of artemisinin. So in late 1960s, malarial parasite developed severe resistance to the standard global drug. During that time, the standard global drug is quinine. The Chinese government, they launched the project they call as a project 523, to find the answer. So one of the researchers, Tu Yu, they tried to look back on ancient medical compendium, this traditional China medicine compendium, and found 1,600-year-old text. So recommending one plant they call as sweet wormwood. So where this plant is used for fever. So however, they tried to see during that time, they tried to study the plant. The modern laboratory tried have attempted to extract an active drug from this plant, but they keep failing. So Tu Yu, they try to return to that text, and then see what the scientists have been conducted to the plant. And then this researcher noticed a crucial detail, where the ancient physician, so instructed soaking the herbs in a cold water rather than boiling it. So Tu Yu, she realized that standard modern hot solvent extraction method destroying the active compound. So by changing the extraction process to a low temperature extraction, so she successfully isolated artemisinin. And then as we know now, the artemisinin combination therapy are the primary global defense against malaria. So this discovery earned the Tu Yu Nobel Prize in 2015. And then so this they become as a master class in reading ancient data with a modern biological lens. And then now artemisinin, because the isolate from the leaves, during the process of isolation, there is a chlorophyll. So the chlorophyll actually is disturbed, the process of the isolation of artemisinin. So some scientists they try to this one a genetic engineer try to copy and then produce this artemisinin in the hairy root. In hairy root there is no chlorophyll. So this will make the scientists easy to purify the artemisinin. Okay, next slide, please. Okay, so the second case, previous is about the infectious disease. Now we're moving to the oncology. Okay, so let's talk about the how natural product integration re-engineered cancer therapeutic. Yeah, so now in the market, there is a paclitaxel, and so this commercially known as a Taxol. Yeah, so originally this paclitaxel discovered through a screening program, that inspired by folklore, uses of the U3 family, or Pacific EU. Okay, so it did not just become a premier drug, for breast and ovarian cancer. Yeah, so it actually revealed an entirely new mechanism biological action. Yeah, so there's for the traditional use, this actually have been used, for example, to treat the inflammation, by local people. Yeah? So with the new technology, the researcher discovered the new mechanism of biological action of this compound, or paclitaxel or Taxol. Yeah. So how this compound frees the cancer cell unit division. By binding. The compound bind to the structural microtubule. So this one of the mechanism of action of the paclitaxel or Taxol. Okay, this similarly to the film that we can find in the Madagascar periwinkle. So this also have been used traditional, as a remedy. So and then after the discovery, or after the scientists discover or isolate the finca alkaloid, fincristin and vinblastin. So this compound transform acute lymphoblastic leukemia in children, from an absolute death sentence into a disease with the cure rate can exceed to 90% today. So this is one of the successful story about finca alkaloid. Okay, so so here, these are not just marginal therapy. They are foundation, for the modern oncology. Okay, so next slide, please. So the third cases that we can see how actually the traditional, when we merge with the technology, this can provide a good therapeutic product. Okay. So this become our final case study. So from forest into the ocean. So this one moved to the marine biotechnology. For centuries, coastal and island indigenous community have turned to the sea. They utilize specific seaweed or macroalgae or microalgae or any marine resources or organism to treat severe infection and wounds. So these have been used by the people, especially in coastal and island indigenous community. Okay, so for modern biotechnology scale, so this traditional ecology knowledge, so they not just stop to the seaweed or macroalgae. So this we need to dive into a deep ocean. A lot of organisms that living in high pressure, low oxygen and toxic environment, for example, like sponges, or extremophilic bacteria. So where these organisms, they cannot run away from predators. Instead, they have evolved highly complex chemical defense system. Okay, so for how they can survive, how they can evolve, to produce a good defense system, they produce a complex chemical. And then some of complex chemical, so these have been used, scientists have isolated and then characterized and then test. Even this can use for the treatment several diseases. For example, as a painkiller. Okay, so here, so by combining the coastal inside with the deep sea, so now, we can see the scientists, especially the marine scientists, they they produce the deep sea robotic sampling. Because we need the deep sea organism to see how they can survive with this condition. Okay, so and then with the sampling, and then the scientists can produce the genomic sequence. We can unlock the entire new pipeline of novel, for example, antimicrobial peptide, and then also peptide that can be used for the treatment of cancer. So this that we can discover. Okay, so the ocean is proving to be our richest repository, for fighting the modern crisis for antibiotic resistant superbug. A lot of organisms in the deep sea still not studied yet. Okay, so this is what we need to see the prospect of this marine organism. Okay, next slide, please. Okay, so in these three cases of study, actually there is a lot of study, just to show you how the medicine that we use now, or the medicine that available in the market now, actually is come from the traditional medicine. Okay, so but from these three cases, so this teach us, it is that traditional knowledge should never be viewed as an alternative or as an adversary to modern pharmaceutical research. This is not just alternative or adversary. So actually, the traditional medicine is an accelerator. It is highly accurate map. So because the traditional medicine have been used since ancient time by our ancestors, and then until now we still use that. So we can see this traditional medicine. This is not just alternative. This accelerator. It is a highly accurate map. When we value the empirical clues left by history, isolate them with modern technique precision, and then scale them ethically, we create medical intervention that alter human history. So this is a good thing. So of the traditional medicine, when we use technology to help us, but doing this work from traditional and we use the modern technique, so this will open up massive ethical, legal and legal battleground. So this will bring us to the next section, actually that I will explain. So on the challenges of intellectual properties and biopiracy. Okay, next slide. So we have looked at the incredible scientific and clinical breakthrough. So where this made possible by integrating biotechnology with the traditional knowledge. But this combination, the traditional knowledge and the technique, modern technique, so this is not always smooth. So there is an issue, when we try to move forward, to produce the product. Okay, so for example, like a critical, ethical, legal and socio-economic barrier. So this will rise during this process, because after we find this extract is good, for the treatment, for example, treatment dementia and so on. Okay, so what next? The next is to produce. Okay, so here, but we need to see the from the angle of ethical, legal, and socio-economic. So we need to know the source, how much of the source that we have, what we need to do, and so on. Okay, so when modern pharmaceutical framework, so which are highly capitalized, private, and profit-driven, interact with the knowledge, the traditional knowledge system, which are communal, ancestral, and sacred. So there is a friction here. So let's look at legal and ethical boundaries that we must establish to ensure this integration is fair and sustainable. Yes, I will explain for the next slide. Okay, next slide. Okay, so the dark side of this field, so it's the phenomenon known as biopiracy. So biopiracy, so this defined as a commercial exploitation of indigenous biochemical or genetic material without authorization or fair compensation. So this is what we call as biopiracy. This occurs when corporations extract genetic resources or traditional knowledge from developed nations, patent them in the West, and generate billions of dollars without the permission of or maybe compensation to the indigenous custodian. So this is what happened now. Okay, so so for example, the textbook, we can say as a textbook example, this Hoodia cactus, Hoodia cactus, So Western farmer, patented this this one actually about the process of the discovery on the Hoodia cactus. The Western people, the people in the Kalahari Desert, so why the Hoodia cactus available in the desert. So this is actually the people there, use this cactus for centuries to starve of hunger during long hunts. So, and then Western company patents its active molecule for multi-billion-dollar weight loss drug. Before a massive legal backlash, force an equity agreement. So this is the condition for the Hoodia cactus case. So to prevent this condition happen again, so the international community established one protocol we call as Nagoya Protocol. This legally binding treaty mandates access and benefit sharing, or we call as ABS. It states that before a scientist even pick of a leaf in another country, they must obtain prior informed consent from the community and sign an agreement ensuring that if a drug is successfully developed, a fair share of the profit and technology flows directly back to the community. So this what decided in the Nagoya Protocol. Okay, next slide, please. Okay, so this from the Nagoya Protocol brings us to the fundamental systemic conflict. Systemic conflict. So there is what we call as the intellectual property dilemma. So between the modern patent law and the traditional knowledge system. Okay, modern patent law, were built during the Industrial Revolution to protect distinct individual timestamp invention. Because the research is conducted by group. So they have their own research on this. This more on the individual. So this more focus. So understand that Western patent law, we cannot patent a natural product directly because it lacks the novelty. We cannot patent the structure. So, but drug company frequently tweak the molecule slightly to the tweak the molecule slightly to secure the patent. Okay, so this will create the massive ethical friction. Indigenous or traditional community ask how can a corporation claim a private monopoly over a healing mechanism that our ancestors discovered and share for the common good. So this the situation when you see this table. So this actually the friction between the Western patent law and the traditional knowledge system. So where the traditional knowledge system, they're more focused on communal. Communal. Okay, next please. Next slide. Okay. So this the barrier. So this the legal barrier. So the barrier is not just illegal. Also the you can see here. So the we need to see deeply in terms of ecological and socio-economic. When modern biotechnology validate a traditional remedy, global demand hits an absolute fever pitch overnight. Suddenly, a plant that has sustainable harvested by a few local healer is subjected to intense industrial supply chain. So we can see how the exploitation of this plant. So this will. There is a tragic socio-economic ripple. So we will lose. There is we will lose a lot of plant, or maybe become endangered species. So here the problem here. So the low supply. For the global pharma consume. Okay, next slide, please. Okay, so for the future, the natural product research here, especially for the biotechnology, this cannot be built on an extractive model. We cannot simply take, tweak, and patent. We need to moving forward. The pharmaceutical industry must champion a co-creative paradigm. This means honoring the legal protection of the Nagoya Protocol, creating a new legal framework that recognizes communal intellectual properties, and heavily investing in the synthetic biology or local sustainable cultivation to protect wild ecosystem. Only by respecting the human ecology sources of our inspiration can we ethically advance global medicine. So next we will look for what the ultimate destination of this integration. Okay, next slide, please. Okay, so this the future, the multi-target therapeutic. Okay, so we now arrive for our final destination, we can say the future. Up to this point, modern Western pharmacology has operated under a very strict, highly successful, but ultimate limited dogma. One drug, one target, one disease. So we look for a single broken molecular switch. We design a synthetic result to hit it. But the most pressing healthcare crisis of our time, the disease like Alzheimer, type 2 diabetes, and cancer, they don't care about single switch. They are highly adaptive. So they have involved complex network. So we need the multi-target therapeutic, where advanced biotechnology is finally learning to mimic the inherently holistic multiple compound design that traditional medicine has utilized for millennia. So the future trying we shift to the multi-target therapeutic. Okay, next slide, please. Okay, so when we are hitting a wall, just using a single target. Okay? So now we move to the several target. With one key, So when you see here, this is a traditional one key one drug for one disease, through the network pharmacology, So we can have the multiple target. A traditional herbal formula doesn't just throw one whistle. Chemical bomb, but when we see in the extract, there is several compounds. So with the several compounds, they can target, they can have a multiple target. So this idea actually is similar to our traditional medicine. We use extract with the complex of the chemical compound, and then when we use the biotechnology technique, so we can provide the drug, the combination of drug that can target several target of action. Okay, so this concept is similar like our traditional medicine. Okay, so this is what we call as network pharmacology. Network pharmacology. Okay, the next slide, please. Okay, so here the network pharmacology approach will unlock the incredible synergy with another huge trend, personalized medicine. If you look at the traditional Chinese medicine or Ayurveda, practitioner will never give the exact same herbal mixture to 10 people with the same symptom. They will provide a different, different, maybe same herbs, but different amount for each herb for different people. They meticulously evaluated the patient individual constitution and then metabolic phenotype. So in modern medicine, is trying to do the exact same thing. Right now, by using genomic, proteomic, and big data. So this feature rely in the precision botany. By combining modern genomic sequence with the traditional constitutional medicine framework. So we will soon be able to print or prescribe multi-component standardized natural product therapy uniquely tailored to a single patient biological profile. So with the metabolomic and AI, so this will help the researcher. So it is the ultimate measure of the of personalized ancient wisdom and personal digital health. Okay, next slide. So this is actually the last slide. So to conclude our journey this afternoon, let's look at the full loop of innovation that we have traveled. We begin with in the forest, grounded in the deep empirical observation of ethnobotany. We cross the technology bridge, seeing how robotic AI and synthetic biology allow us to decode, to standardize, and to scale this discovery without destroying our ecosystem. We navigate the vital ethical and legal guidelines required to protect indigenous intellectual property. And finally, we look at the future where pharmacology learn to think holistically again through the network therapeutics. So the takeaway from this, the future of medicine does not require us to abandon our past. So the next generation of the global pharmaceutical breakthrough is quietly waiting for us in the molecular complexity of our oldest tradition. Okay, so by protecting our planet's biodiversity, honoring its human custodian, and deploying our most advanced technological tool, we can build a safer, more sustainable, and truly integrated future of global healthcare. Okay, so that's all from me. Thank you. I pass back to the moderator.
Thank you. Please give a warm applause from Mrs. Danny. It was spent a whole thing very complete, a holistic explanation from the history of traditional medicine. Then many plants have transformed into the medicine until the AI thing that very up to date nowadays. And then you explain about the alternative such as marine biotechnology and then deep sea bioprocess that has a great prospect and opportunity to researcher. Then the barrier and issue in this field and also the future multi-target therapeutic. So if you have a question, please keep it. You can ask after the second presentation. And thank you, Mrs. Danny. We kindly ask you to stay in the Zoom meeting for the Q&A session following the second presentation.
Okay, thank you.
Okay, now let me, is there Professor Taher already joined the Zoom?
Yes.
Okay, good afternoon, Professor.
Good afternoon.
Okay, now it's my great pleasure to introduce our second speaker. We are honored to welcome Professor Muhammad Taher bin Bakhtiar from the Kuliah of Farmasi, International Islamic University, Malaysia. Professor Taher obtained his master's degree in organic chemistry from University Technology Malaysia in 2001 and completed his PhD in bioprocess at the same university in 2005. He has extensive experience in pharmaceutical biotechnology, natural product research, and drug discovery. Today, Professor Taher will deliver a presentation entitled Pharmaceutical Biotechnology in Natural Product Drug Discovery. It's my great pleasure to invite Professor Muhammad Taher to deliver his presentation. Professor, the floor is yours.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much, Madam Moderator. And first of all, I'd like to thank Fakultas Farmasi Universitas Pancasila for having me in this yearly event, Farmasi's International Pharmaceutical Summer Camp. For today's presentation, I would like to share with all of participants and other guests perhaps attending this camp what called physically, yeah. So I will elaborate through Zoom. So allow me to share my slide. Is it visible for everyone?
Yes.
Okay, thank you. So basically my topic is actually to add on to the topic what actually Dr. Rani had presented. As we know, natural product is one of the source of modern medicine today. And we know that the process of drug discovery and development takes quite some time and costs about million or billion US dollar. And actually, as alternative how to make the process will be faster, or how to make how to overcome the additional hindrance or problem. So we can do the approach, through biotechnology product, or a biotechnology process, and alternatives can be using AI later on. But today our topic is actually using biotechnology, and enhancing, to speed up the process of production. So, and as an overview of my today presentation will be covered from the general overview regarding the natural medicinal products, started from discovery platform and also foundation, to understand the core component and role of biotechnology in modern bioprospecting. Let's look at the nature provide everything to human life, especially in pharmaceutical field, and how to treat disease. People have called deep their effort actually to find how to make a healthy life, how to treat a disease, example. As you know, there are many sources of natural products or natural medicine. This can be from plants, fungi, marine organism, also microbes. And as we know, the process of development of natural products from long time ago until today, it has been called change from traditional ways to modern ways. Basically, the objective is actually to speed up the process, and to reduce the cost. I mean, what we do is actually doing efficiency in the process of the production. As we know that actually about 50 to 70% of approved drug nowadays are derived from the natural products. If we look back to the traditional way how to produce natural products, right? That actually we know the process will take time and also costly. A little bit for your brief information regarding the process, we have to find the source through server or maybe through botanical what called question, something like that. And then after that we do extraction. And then after extraction, what we need to do is actually doing isolation and purification. And then after that we need to establish the structure. After that, what next is bacteria screening, and then we'll find lead compound. I mean, it is still a big question. What compound is it? And then this process is a very lengthy process. Lengthy process, maybe through generation as well. Let's say if we are doing in the linguistic sample, maybe we have already changed the student from undergraduate, postgraduate, master, PhD, maybe it's professor also changed, but they're still not finished. And then you see why it takes time, or what is the major bottlenecks or actually problem in this traditional ways, because of difficult in isolation. And then if we try to extract compound from the plant sample, very difficult to make it pure through isolation. Of course, we have to undergo several steps of chromatography in order to get pure compound. It will take time as well. And then the problem is low yields. We try to actually get, for example, new compound. Normally, the new compound is actually minor compound. So the compound is very low in terms of yield and the extract. So becoming problem as well, because we want to sample to test it in assay sample, or want to do some kind of development or formulation, become a problem. And then try to rediscovery of non-compound, maybe perhaps you can get it from through chemo, what called came diversity, yeah. I mean, from the same genus or maybe from some same family, yeah. And also the time of the process, yeah, cost, yeah, and also related to supply and reproducibility issues becoming a problem, because perhaps many factors actually regarding the supply and reproducibility issues. For example, geographical age of the plant sample, yeah, and altitude, yeah, also play quite important factors, yeah, in order to get the same compound, yeah, from the same plant. And then you see the problem. I mean, scientists try to improve it, yeah, from traditional ways into modern ways, yeah. Maybe how actually the technology play their role in this drug discovery. Okay, I can show you in it can be using genomic, yeah, doing genetic modification, yeah. Or other thing is using biotransformation, using living organism to change the structures, yeah. That normally we do it in the lab using chemicals, with chemicals, and change the structure to get new compound. Or in this case, we use microbial metabolites, enzyme, actually, to change the compound or improve the compound. Or maybe through callus culture. Callus culture, we can do it in the lab, to produce certain active compound in the lab only. No need to do plantation, field plantation, because perhaps I mean it is not controlled. Yeah, we can actually doing this production to under controlled environment. And then the technology will change the game by doing this kind of impact, faster identification, smarter screening, and reduce rediscovery, scalable production, and precision optimization. So all of these changes actually it's not affordable. It's not available in traditional, traditional discovery. What can you say? Yeah, traditional discovery. Then what next is actually the general information regarding this topic is genomic and metagenomic. And understanding regarding what the process of transformation from gene to protein, for example. What we are familiar is with DNA, is with called recombinant DNA technology. Yeah? And this actually in this drug discovery we can also apply the same method, from the biosynthetic gene cluster. I can see this to gene actually to produce drug heat. Of course, through what called living organism, doing it for us. Not just depend on what source can, what natural can give us. And also we can use something actually what we actually expect from natural. Through this, control, or maybe through this technology, knowledge and technology, in order actually to get what we want, actually. Not only what natural can give us. We can do kind of actually prediction. Of course, later actually the successfulness is not only belong to God. But we try our best actually how to design it. How to design it. And then understanding what called the use of gene using CRISPR-Cas9 is actually a gene modification tool. Understanding this how this CRISPR-Cas9 works. in order to modify the gene, yeah, add a gene or cut the gene, strain gene. Yeah. But actually, the idea is actually do it from inside. Yeah. We do kind of modification before production. Yeah. Not like you do kind of modification of compound, for example, that one is after the product and the compound is already produced. Yeah. But using this Cas9, actually we modify the gene of certain gene, yeah, in order to promote, yeah, the production of certain compound, for example. Similar to what we do in recombinant technology, let's say you want to produce insulin. Yeah. For example, we have to cut insulin gene, yeah, combined with plasmid, something like that. Yeah. So then the plasmid will be transformed to a microorganism like E. coli, and then the E. coli will able to actually to translate, yeah, the gene that already inserted into plasmid to produce a length of protein, we call it insulin. Yeah. Similar process happen when we want to, but perhaps it's not truly like what we do in recombinant DNA technology. Yeah. But gene modification here is play the role, yeah, to produce certain compound. And then after that, of course, we can compare the metabolic profiles, yeah, of the perhaps non-compound with the product that has been produced through this genetic modification. And then other method, the updated method actually in drug discovery, yeah, in addition to biotechnology, what I can, this is very important nowadays, using AI, LLM, yeah, large language model to improve, because the data means something that can actually transform to AI, yeah. Sample, yeah, the natural product data input. As we know, the molecular structure sometimes quite, this skeleton, right? I mean, similar structure, and later we can actually try to make, yeah, or pattern the structure, yeah, and then after that we can use AI or deep learning engine, yeah, and then we will predict regarding the structure and scaffold, yeah, biological function, drug likeness, and also novelty and characterization, yeah, in this kind of activity. Yeah. So meaning we can do it before, yeah? And then after that we can, I mean, it is like docking things, yeah? But this I maybe because it's the latest, yeah. What it has been done before using molecular docking something, and before we go to the lab. So meaning we have already get some idea. Sometimes I want to do synthesis, yeah. Before we do it in wet lab, we have kind of guidance, yeah. So maybe the process later can be faster, yeah, than compared, yeah, to the thing that we don't do kind of simulation first. Yeah? Okay. Then move to the steps, yeah, and perhaps in details how actually we can implement, yeah, these ways actually to produce natural products. So started from natural architectures, establish why biological natural products are critical asset, therapeutic innovation, drug design, and also modern molecular architecture pipelines. Meaning we have to appreciate, to acknowledge that natural product is actually sourced, perhaps not only source in terms of physical product, also a source of idea as well. Source of idea on how actually we can improve what natural can give us. Not only depend on that, maybe with the knowledge we can actually modify. We can do something to improve the product. And then through that biotech innovation, in order to to improve the process, speed up the process, and to improve the yield sample, and then Last step is regarding the perspective, yeah, to analyze the key case study, yeah, addressing scale, bottlenecks, and so on. Yeah. And little bit regarding the extraction process and synthetic biology. I mean, if we looks, yeah, how actually the improve process like the technology can help, yeah, us in order to speed up and can to work about to overcome the problem. Yeah? In synthesis extraction, we have to depend on the resource, yeah? Because resource also depleted, yeah? Massive what called massive harvesting sample can deplete the sample, yeah? And extreme supply chains, yeah, through the geopolitical conflicts, like war, yeah, can also affect the the traditional ways of drug discovery, yeah? And healthy solvent content chemical, because the process is consuming or using chemical solvent, yeah, it can actually make the product to be polluted, yeah. As we know, that one can be actually removed to be get rid from the product, yeah. And you can see the sustainability is actually to improve the production through zero environmental footprints, yeah, and stable year-round production. Nothing, I mean, there is no problem with the supply of sample, yeah, and also scalable biosynthesis, yeah, directly produce ready to purify molecules, yeah, because in the production using synthetic biology, yeah, perhaps the impurities will be less because we have already designed from the beginning. Yeah, the perhaps in the end the product may be little bit contaminated by minor compound. Yeah, for those who are familiar with the extraction, yeah? When you see the component of the extract can be 100,000 compounds there. Yeah? But through synthetic biology, perhaps the product is not really can say contaminated by other compound. Yeah? Okay. And then what actually can help us in this process of transformation is actually using microbial cell factories. Yeah? For those who are familiar with microbes, right? So microbes actually can be used as machinery, yeah? In order to produce compound or maybe protein, perhaps protein, of course. Yeah? So this actually one of the what call it the idea to produce natural product, yeah, other than from direct extraction from plants. Yeah? And then the target molecules process, yeah, what is the compound target? Of course, when we are dealing with the drug, you know, we have to know what class of compound is there, yeah? Whether it is terpenoids, for example, alkaloids, polyphenols, or polypeptides. Yeah? And as you know, all of the chemical class, they have their own representative. For example, terpenoids, you know, artemisinin, yeah, is actually anti-malarial drugs. Yeah? Morphine, alkaloids, yeah? This for analgesic and pain relief, and so on. Yeah? So you have to know what target molecule or class, yeah, would like to produce in this case. And then, of course, when we are dealing with the technology, you have to play with the gene, must be what called familiar with the process of molecular things, yeah? Like what type of gene can be used to cut, to glue, to, you know, like this is actually technology CRISPR-Cas9 is actually gene editing tools, yeah? Actually can help us, yeah, to cut. Yeah, understanding this concept, perhaps we can open our mind. Actually, we can do many things. Actually, I mean, based on the small things in our life, right? Through gene modification, yeah? In order to produce whatever we want, yeah, you can actually, make some kind of idea, yeah, to utilize gene in the process of production. And then you see the application of CRISPR-Cas9 technology for genomic editing has been used, yeah, not only for drug discovery, have been used in biology, yeah, and engineered animals, biotechnology, biofuel production, plant technology, as well as in medicine. Yeah? So this actually not really new. Perhaps because we are doing production, maybe perhaps a little bit specific, yeah, to the production of drug sample. Yeah? So not just limited on this drug discovery. Yeah? It has been used in many disciplines as well. Okay, and then again, why natural products? Not actually to be asked, yeah, but just to again, yeah, to open our again regarding how, yeah, important natural product to our life, yeah, specifically for medicine, yeah, because as we know that 50% of approved drugs nowadays inspired or maybe isolated from plants, yeah, like from natural products, yeah, can be from plants, microorganisms, bacteria, fungi, and also microalgae, yeah, that they have been used in oncology, yeah, antimicrobe, metabolic control, and also anti-inflammatory. And again, regarding the classification of natural products, perhaps you know about this, yeah, like it can be alkaloids, yeah, example morphine, yeah, saponins, yeah, terpenoids, flavonoids, and also phenols. All of them are important, yeah, in our life, yeah, whether it has been used for prevention, yeah, or maybe in the treatment, yeah, of disease. Yeah. Then regarding the source of natural products, this again is maybe repetition, yeah, in the previous slides. Yeah. So then natural products, yeah, meaning from natural. Yeah. What can what can actually produce medicine for us, right? So it can be from plants, animal resource, microbial resource, and also mineral resources. Okay, and the application of natural product itself, yeah, as we know, natural product is very broad. Yeah, they are not only for drug that has been utilized by a pharma company. Yeah, also can be used as a crop. Yeah. In fragrance, volatile chemical compounds, and food additives, color, wood and paper, plastic, and also for industrial catalysts. Yeah. So perhaps natural product. Talking about natural products, the percentage of drug in there is very small, perhaps about from 20 or 15% of the total use of natural product, just only for drug or medicine. Yeah. And the role of technology in production is actually can be classified into two, in discovery and analytics, and also in production and engineering. Yeah, discovery and analytics is like in multi-omics integration. Yeah, I target identification, also high throughput screening. Yeah, and next process is production and engineering. Yeah, and synthetic biology, CRISPR-Cas engineering, and also recombinant proteins if the product is related to proteins. Yeah. So, so the question is whether the insulin, that has been produced through recombinant technologies can be called as natural product, and this is a big question. Whether it is considered as natural product or not, because their status perhaps is actually through source is from human sample or from animal, actually already joining with the plasmid and whether the product is considered, but they are under protein product. Yeah? They are under protein product, but whether we can call them as natural products. Yeah? And scientific advances and case studies, yeah, little bit we see the how the technology has been changed the process of drug discovery and development. Yeah? Then the big question is why technology become an option, because of the source of plant is difficult to cultivate some. Yeah? Maybe the plant is very rare to be cultivated, so we can do it through technology. And it has long cultivation period, the timing to get actually matured, for example, quinine. For example, you want to isolate the quinine from quina, you have to plant them, and many other sample. And then it has low metabolic yield. Metabolic is very low in terms of the like what happened right now in the production of vincristine, from Finca rosea plants. The yield is very low. I think this what call it, this vincristine need to try alternative way how to produce it through technology. And chemical synthesis has not been achieved due to technical problem. So, as you know, the process of the production can be through natural extraction, or we do it in synthesis in the lab, chemical and chemical. Maybe this chemical synthesis has not been achieved due to the technical problem sample, or maybe difficulties in terms of synthesis, because the structure is very complex. And then novel compound which are not generally found in the parent plants. So the compound is not from there. Perhaps, of course, the study must be comprehensive regarding what target or compound need to be obtained sample. And also, sterile and region-specific preservation of plant cells can be performed for production of reactive compound. Then, we will talk about biotransformational natural products. You can actually just for your brief information through plant-derived metabolites or through reaction, less and also enzymatic mechanism. It's actually using living organism to transform the compound. Not using any catalyst. The catalyst is actually the metabolite or enzyme that been produced by microorganism and help us to do something on our compound. Example, the compound is very common. Example, you want to get new compound, maybe you already not successful in synthesis, or maybe not successful in chemical modification. We can use living organism or microbes to do kind of transform. Transform here is just to change a little bit about the substitution. So this the process, based on the journal that we actually read. So the biotransformation can be done by bacteria or fungi. So the compound, we take the compound, and then to culture it with microbes, and after that we extract, and of course next is using the common process like chromatography, aspiration and structural identification. This actually the study that has been done on ginseng. They use this kind of called the technology in order to produce compound from ginseng. So they use cell factories in order to produce the compound from ginseng. Then other method is using plant tissue culture. So as you know, the plant tissue culture, one of the perhaps it's not a very successful one, but an attempt has been done. Yeah? Ideal people will say that we can culture them actually in the lab, but the process is not just simple like that. Yeah? But it was successful for certain compound. Maybe not applicable for all plants, because some plants very easy to be cultured in the lab. Some plants may be very difficult. Sometimes cannot be cultured in the lab. But this kind of effort, I mean, alternative in order to produce active compound from plant. And this is the call it they use callus culture. The plant stem cell, they try to grow in callus culture, try to to grow them, and later perhaps you can actually do kind of gene transformation here. Gene transformation to the cells, then you produce certain compound, I mean target compound in the product. This is the process of flow and technology transfer and plant culture. We do seed trains, including train, production, and harvest. This is the process how actually we can use technology, technology process in order to produce. If you're familiar with the community technology production, almost the same process, almost the same process. But perhaps this one is need to be done in specific bioreactor in order to produce certain target compound. And the current scientific advances, of course, through BGC mining just now, through precision fermentation and single cell proteomics and nanotech-based drug delivery. This is the current scientific advance that can actually be used to produce alternative natural product production. And a bit regarding the successful case studies, like this ladies is very familiar face, right? And the production of artemisinin. Artemisinin is actually from artemisia that can be used to treat malaria. This lady's got a Nobel Prize. I forgot the years. And then the production of Taxol. So this one is actually in according the process. The production, because we know that Taxol is isolated directly from taxus plant. So try to produce it through culture, and actually from marine oncology also can be one of the strategy actually, because of marine difficulties in terms of the harvesting, and then there is a site that can actually to collect the raw material, so I can do kind of alternative, because marine, as you know, that marine product is very rich in anti-cancer drug. So try to do kind of alternative ways in order to produce the active compound. Again, this one is case study. Can see here the internal products is artemisinin, dextroadenosine, and also marine metabolites. That's actually the explanation of a few slides. And overcoming core bottlenecks, the problem here actually how to solve the problem. It is regarding resource scarcity, the limited source, and then to solve a problem about a complex isolation. You see the natural, the traditional extraction, you can get any compound that extracts, so the extraction will be complex. And also regulatory, as Anthony mentioned just now, I think it's related also with the requirement by global regulatory body like FDA and EMA. And regarding biodiversity integrity, this one is regarding the call what call that can affect the biodiversity. If you explore it massively, example, will affect the environment, diversity. And then for future horizons is using digital tech and precision, biomanufacturing. Not only precision here is not only precision medicine and also precision in biomanufacturing. Can use AI with multi-precision major shift, enabling digital prediction of several drug activity before running real-world trials. And this what called future, precision manufacturing is customized horse. Environments allows the biopharma industry to produce clean, infinitely scalable medicine from extreme, previously unreachable deep sea or thermal cosmetics. As Tech mentioned just now, you say we have to actually make things more safe, make things faster, make things low cost, for example. So usually the benefit that can be obtained from this biotechnology technique. So I think that's all for my presentation today. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. I will give it back to Madam Moderator. Thank you very much.
Thank you very much. Give applause to Professor Taher. Thank you so much for the inspiring explanation. Also, we invite Associate Professor Dr. Jenny Susanti to join us in question and answer session. So, as I mentioned earlier, we will open 3 questions for each speaker. So we have a total 6 special gift for who want to ask the question. So who would like to be our brave participant? Okay. Please introduce your name and maybe you can face the camera and say hi to speaker. Or maybe we, the committee can move around the camera so the speaker can see the participant. Look the camera, okay? And say hi to speaker and mention.
Good afternoon. Thank you for the opportunity to Professor Taher, Professor Susanti, and Mrs. Naya. My name is Natania Felicia Wijaya from Pancasila University. I want to ask to Professor Susanti. So with the rapid advancement of AI in drug discovery, do you think AI will eventually become capable of identifying promising medicinal plants without relying on traditional knowledge, or will ethnobotanical knowledge always remain an essential component of bioprospecting? Thank you.
Okay, thank you. Mrs. Denny.
Okay, so
Okay.
okay, so I just answer directly the question. Hello.
Hello.
Okay. So the question, yeah, so about AI, whether AI can is it AI can translate or can predict the activity that that we can see from the traditional medicine. Okay, so AI is the AI that we can use is a machine learning. Yeah, machine learning. So this will capture. Yeah, this will capture all the information that we provide them. Okay, so for example, like I mentioned, we need to provide AI with the data we get from metabolomic. Yeah, so this is a big data from metabolomic. So we know what is the extract. Yeah, so the component in the extract. If in the extract there is a 10 or 1,000 or maybe and maybe hundreds of components responsible for the action. Yeah, so we know from metabolomic. Okay, so and then through the HTS high throughput screening, yeah. So where we test, this extract, to the target. Yeah, the target, for example, maybe to the cancer cell, to the protein. Okay, so we do the screening on that. So there is a result from this. Yeah, so whether this extract that consists of the bioactive compound, have ability, for example, to inhibit the growth of cancer cell. So there is a data for that, through the screening. Okay, so this data we we give input to the AI. Okay, so for example, if they inhibit certain protein, for example, so we have so this is a metabolomic study that we can produce. Okay, so we put in AI. So AI will see we see the data where the data that responsible, for example, the protein is inhibited and so on. They have this on. So the AI will predict. AI only predict. So with this prediction, so we can scale down, our research to prove it, biologically. AI, AI and metabolomic is just a bridge. So until we find, so from the data that we get from metabolomic and then metabolomic, this not only the list of chemical, but the research that we conducted, we test for biological activity. So this also data. And then AI will see, there is algorithm, they see this data and then predict the action. AI will help us to shorten the process of the discovery. Okay, so for to prove this, we still need biological testing. We still preclinical to prove it. We cannot just pass AI and metabolomic to shorten the process. I think that's all. Am I answering your question?
Yes, Professor, thank you.
Okay, thank you. So AI can help us to predict and make more efficient, I think. Okay. Next, please raise your hand. Anyone? Please, I think everyone thinking the perfect question. Okay, please, from Tua University. You can look the camera.
Okay. Good afternoon, Professor Danny and Professor Muhammad. I have a question for Professor Muhammad for his lecture earlier. So, I'm Nicasia De Leon from Trinity University of Asia, and you mentioned that CRISPR-Cas9 can be used to activate silent biosynthetic gene clusters. My question is, once these silent pathways are activated, how often do researchers discover completely novel bioactive compounds rather than just increasing the yield of
Okay, thank you very much for the question. Very hard question to be answered. If I actually can get the question, the first one is regarding the power of CRISPR, yeah, in order to uh, suppress something. I mean, the role of CRISPR in the production of secondary metabolite, right? So, yes, this kind of still new in the field. So what can this actually to give an idea to us on how to find alternative ways, yeah, in the production of natural products, and not only depend on traditional extraction. Yeah, but honestly, I cannot answer exact, because not many works has been have been published, yeah, through this technique, yeah. But it is very promising ways actually when we try to produce natural products. I mean, we can, because the production of compounds normally through what called genetic, yeah, process, yeah. Gene is very important here, yeah, in order to produce, to suppress something like that, yeah. So perhaps we are together still need to learn, yeah, and because not too much information that has been published, yeah. So my answer is actually this one is very what called I can say is promising, yeah, and also can be used in alternative ways, yeah, in the production of natural products. And second question, if I may, dilaporkan Madam actually again, but can you recall the question? The third one. Okay. Uh, the main challenges is actually if you are familiar with recombinant DNA technology, right? I mean, it is almost similar. Yeah. So the main challenges in is actually actually to find the what called to cut, yeah, and also to again to glue the gene, yeah. So the main process is on molecular part, yeah. The main challenges is in molecular part. Once you are successful, in dealing with the molecular things, yeah, the process will be much easier, yeah. Yeah. So yes, actually we can target what actually I would like to produce through genetic sequences. Yeah, but this molecular part is very challenging. Yeah, what I mean in terms of line of the gene, yeah, how to combine them, something like that. Yeah, so this is really the main challenging. Yeah, if I answer your question.
Okay, is it clear enough? Okay, thank you. Anyone else to want to share the question? Dio, maybe? Okay, Mas Ari, help to move the camera. Just move the camera to back.
Thank you for your presentation. First of all, my name Dio. I'm from Pancasila University. I want ask something. I want ask to Mr. Denny. Do you think biotechnology will also help develop personalized medicine from natural product in the future?
Okay.
Okay. Okay, thank you. Biru, yeah. Okay, so I just answer directly, Ms. Moderator.
Okay, please.
Okay. So your question is asking whether the biotechnology, yeah, can provide the personalized medicine. Okay. Okay. So through biotechnology. Okay. So biotechnology, yeah. So this become a bridge, yeah, for us, yeah. So we can go to the personalized medicine. Okay, so why I say that this become bridge, because of the through the biotechnology process, yeah. So we will know, yeah, everything about the compound, the responsible, this compound maybe not only one, but the mixture of the compound. Yes, so we know, and then also we also can predict, yeah, the action, yeah, of this compound, yeah, through the biotechnology process. Yeah, indirectly, yes, this can help us, yeah, to go to the personalized medicine indirectly, yeah, because this there is a long process that we need to go. So by like I mentioned, like the traditional practitioner, yeah, especially for the traditional Chinese medicine, when we see them, yeah, so maybe we have a similar symptom, yeah, so we have, for example, we have fever, yeah, okay, so the practitioner, yeah, like I mentioned in the presentation, the practitioner will provide us with the same herbs, with the same amount of each herbs. Yeah? So usually practitioner will have a data about us, everything about us. So there maybe, yeah, for me, so the practitioner will reduce herbs A, or maybe for you this will increase herbs A. So this what we call as the personalized medicine in terms of the traditional. Okay, so in pharmaceutical, yeah, in the modern medicine now, so we are going toward that, yeah, personalized, yeah. So this because even DNA, right? So we no, even DNA is we personalized medicine, yeah, indirectly. So because methodology is just a method, yeah, just a method to understand, yeah, about the yeah, bio and technology. Yeah, so technology that we play in the bio. So this is just method to help us to go toward the personalized medicine. Can I answer your question?
Okay, it's clear, Biru? Okay, thank you, Mrs. Denny. So
Okay,
the
thank you.
technology is indirectly help us to support personalized medicine. Anyone else to ask? From Tua, maybe? Okay, from Pancasila?
Thank you for the opportunity. So my name is Safira Nangso. I'm from Pancasila University, and I want to ask a question to Professor Susanti. Countries like Indonesia and Malaysia have rich biodiversity. So the question is, what should be the first priority for developing countries to make better use of biotechnology and natural product drug discovery? Thank you.
What should be the first priority?
What what should be the first priority for developing countries to make better use of biotechnology to in natural product drug discovery?
Oh, okay. Is it clear, Mrs. Denny?
Okay, so what is the priority? Priority now, yeah. So I think all the traditional medicine that we have now have been documented, right? So there is I think there is a pharmacopoeia right now in Indonesia for traditional medicine. So we have documented that. Yeah, we have documented that. So with that, so we can start. Yeah, we can start using the metabolomic, yeah, and then AI, yeah, just to screen everything from the data that we have from the traditional medicine. So until once we have the big data and we analyze with AI, yeah, so from this data we already make the data become more concise, yeah, become more yeah, we can see, yeah, the data become more accurate, yeah, from the traditional, yeah, and then for the effect. So this the first thing. So through the metabolomic, yeah, to compare everything, yeah, from the traditional medicine, the metabolomic here. This is not only for the content of the extract. What happen after we test? For example, for cancer cell or for diabetes type 2 testing, what effect after that? So this also metabolomic data. Yeah. So this is the first thing we need to do. Yeah. So to compile all the big data. Yeah, and then after that we screen using AI. So we have we can see from the prediction of the AI. Yeah. So what is the combination of the compound in the extract that responsible, for example, for the Alzheimer, for the type 2 diabetes, or for cancer? Yeah. So from that, so we can proceed, yeah, maybe, yeah, for the preclinical or clinical. So the first thing is to compile all the data. Yeah. So this the important thing I need to do before we go for all the network of pharmacology and so on.
Yes.
Okay, am I answer your question?
Yes, I agree also. Data is everything. Data is a power right now.
Yes.
And then we can speak by data. Okay, anyone? Taher and Mrs. Denny. What skill do you think pharmacy students need to prepare for the future of pharmaceutical biotechnology? Prof. Taher or Mrs. Denny, please. What skill do you think pharmacy students need for the future pharmaceutical biotechnology?
Maybe I answer first, and then Prof. Aher will see from the other perspective later on. Okay, so for me, because I focus on metabolomic, so and AI, so if pharmaceutical student or pharmacy student want, to work on the natural product field, especially to discover, for the future medicine, So you need to learn about metabolomic. A lot of equipment you need to learn how to interpret metabolomic, all the omics, this one maybe metabolomic. There is a lot of omics like proteomic, genomic, so to study, to get the correct data, okay, and then AI. So AI is the with the new technology, with the AI, so AI is not only ChatGPT. Okay, so there is a lot of machine learning that will help us. Will help us. So this the new technology that we can learn. Even you can learn by yourself. No need to put in the curriculum and so on. We can learn by ourselves. So this the knowledge that the pharmacy need for the future. Okay, maybe Prof. Taher will add another knowledge that we need to give to the student, especially pharmacy student.
Okay, thank you very much for the question. So I think as a pharmacy student, as we know that pharmacy is actually applied now. I mean, not fundamental knowledge like what biotechnology student learn. So perhaps in terms of skill in biotechnology, perhaps we don't have enough what called knowledge or background, because it's related to genetic knowledge. But perhaps as pharmacy student, we should aware that biotechnology is very important in the production of biological product, biologic drug like insulin. How important insulin nowadays during the demand, high demand because of diabetic or metabolic disease. So because people nowadays really appreciate the biologic product. What is biologic product? Biologic product is product that normally how the people will produce it. Like insulin, they produce it. Like interferon, the body produce antibody. But if something happens to our body, we need to supply from outside. So to replace or to add on to the current one. So many because as we know, the process of drug discovery and development is not what called lonely. Not single man show. Like what we experienced in COVID-19 is biotechnology field as well, right? Learning how to get mRNA vaccine, RNA from virus to make mRNA vaccine. So how many percentage? Pharmacists play. In that activities, right? Very small, but in terms of the what call it the usage, So in terms of this what call it knowledge, understanding regarding the product, pharmacy should know. But for the skill, perhaps I don't think we have good background or knowledge in. But the skill in terms of the development, yes. Like in development in terms of formulation, for example, we want to make liposome, encapsulated yes. But from the what could be the production part, perhaps not much, yeah. That can be because they are other they are, I mean, the most capable person is there, like biology or microbiologist, something like that. So I mean, what pharmacy students should aware is regarding the use of biotechnology product in medicine. Yeah, that's all. Thank you.
Okay, thank you, Professor Taher and Mrs. Denny. Before we conclude today's session, we would like to present a certificate of appreciation to our distinguished speakers as a token of our gratitude for sharing their valuable knowledge and expertise with us. The certificates will now display on the Zoom screen. First, we would like to present the certificate to Associate Professor Denny Susanti. Give applause for Associate Dr. Denny Susanti. And next, we would like to present the certificate to Professor Muhammad Taher bin Bahtiar. Give applause for Professor Taher.
Thank you very much.
Thank you once again, Professor Taher and Associate Professor Dr. Denny, for your inspiring presentation and for taking the time to be with us today. We truly appreciate your contribution to the third International Summer Course Pharmacy. Thank you, everyone. I hope you enjoy the rest of the International Summer Course in Pharmacy. Have a wonderful day, and MC, the floor is yours.
Once again, thank you, Prof. Denny and Prof. Muhammad Taher. And we also sincerely thank our moderator, Mrs. Naya, for leading today's last material session. Okay, so before we head to the closing session, I would like to give some information to the participant that actively ask some question in third session and the fourth session. So the first participant is Nikeisna De Leon.
Please come to the stage. Come to the front.
Next one, Natania Felicia. And then Dio Muhammad. And the last one, Safira Naswa.
picture.
Yeah.